In this exercise, we're looking at different physical agents that can affect the growth of bacteria. Specifically here, we're going to look at the effects of heat on bacteria. Now, physical agents can be things such as osmotic pressure, ultraviolet light, the heat is a big one. And so some of these physical agents, they can either slow down the growth or completely destroy the growth. And so you use some of these factors to your advantage in terms of controlling the bacterial growth. Obviously, sometimes you're studying things, you want to see the growth, but other times you don't want to have contamination. You don't want them growing in certain things. You want them sterile, or you don't want the pathogenic bacteria to be growing. So we can control the growth by inhibiting, preventing it from growing, or by killing them. And not all the time do you kill the bacteria. Sometimes you're just inhibiting, so if favorable conditions return, they might be able to grow again. In a lab, one of the things we use is an autoclave, and we use this to sterilize. Now, in microbiology, we're very picky about our terms. Sterilization means that you are killing everything in that particular, maybe tubes, whatever you're looking at as far as your samples. You're killing everything in it. If you sanitize something, you are just reducing the number of microorganisms that are there. We talk about reducing the load. It just means you're reducing the number. Hopefully you're killing off the pathogenic ones. So in a lab situation, we often want to sterilize things. We don't want anything growing, so that then we can purposely, say, inoculate a petri dish with the organism that we want. An autoclave works by using pressurized steam to sterilize. Moist heat is often used. You can also use dry heat. Moist heat, such as boiling, the way it's effective at killing microorganisms is that moist heat at a high enough temperature, such as boiling, will denature your proteins. Structure determines function, so if you destroy the structure of the protein, that's what denaturing is doing, is it's going to alter the structure, which means the protein is no longer functioning. If the protein is no longer functioning, that is going to end up ultimately with the death of the cell, because so many of the various chemical processes that are occurring inside the cell, proteins are involved with them, are responsible for them. So if you destroy the proteins, the processes cannot occur. If the chemical reactions cannot occur inside the cell, then the organism is going to die. Now, spores can be released from those organisms that do produce spores. Not all of them do, but some will. They can produce the spores and release those. That's a survival mechanism to allow the organism to survive under harsh conditions, maybe the increased heat. UV light will damage the DNA, and so that's why it is effective also as decreasing the amount of growth. It will kill some cells. Osmotic pressure can also be used to control the growth. Specifically with heat, as I said, you've got moist heat and dry heat. Moist heat in the autoclave, you use moist heat under pressure. Specifically, it needs to be at a minimum of 121 degrees C. It needs to have a minimum of 15 psi, pounds per square inch of pressure, and it needs to be at an absolute minimum of 15 minutes. Usually we go longer than that. The length of time is also going to be one thing that will affect it, or will determine whether the autoclave is effective, is the amount of supplies that you're autoclaving. The larger or increase the amount of supplies, the longer you need to go. Also, something to consider is that the heat has to be able to penetrate through the sample. So let's say you have a flask with a liquid broth in it. If you have just a small flask, maybe 15-20 minutes up at that elevated temperature and pressure will be fine. Let's say we have a flask with only 50 mils of broth in it. If you have a larger flask, say that has 2 liters of volume in it, that's an increased volume. And so you're probably going to have to go a little bit longer on the time for the heat to be able to penetrate deeper into that amount. With moist heat, such as the autoclave, yes, that should sterilize everything. One thing you have to keep in mind is, will the high temperature possibly have an adverse effect on your materials? So some things cannot be autoclaved. So if it's a heat sensitive thing, you're going to have to find another way to sterilize it, to inhibit or destroy growth on it. Dry heat, an example of this is incineration, where you burn things. That's what you're doing every time you flame your loop. The temperature and the time, as I said, are critical as to how long do you go under various circumstances. Always keep in mind the endospores are more resistant to heat than the vegetative cells. It's a survival mechanism on there. So years ago when I was doing research, as an example, we were trying to sterilize soil. We needed it sterile, so we would take it, we would autoclave it. The first round through the autoclave we knew would be killing the vegetative cells because they're more susceptible to the heat, but the endospores might have survived. So what we would do, we would autoclave the soil, we would take it out of the autoclave, keep it closed. We usually worked with containers that had aluminum foil over it, so we would not open the containers to expose them to the regular air in the room. We kept everything sealed. And then 24 hours later, we would run it through the autoclave a second time. That way, in case there were endospores, the first time round that might have survived, it would have killed the vegetative cells, but the endospores might have survived. Then 24 hours later, when we ran the samples through the autoclave a second time, then it would kill those endospores. In between would have germinated into new vegetative cells. So the second time around, they got killed. And then sometimes we would autoclave it a third time, just kind of for good measure. So it depends on what you're doing, what your samples are, as to what method you're going to use. But heat is a very effective way of acting as a physical agent that is going to control the microbial growth as far as inhibiting it or killing and preventing it from growing in whatever environment you're in.